Hi, so in this lecture I'm going to talk about various network properties that researchers identified in various types of biological networks. So in a review article that was published in 2009, I identified some of those general topological properties that people have identified in various types of biological regulatory networks. These properties include the power law connectivity distribution that we discussed, party hubs and data hubs, multi-site and single-site hubs, power law distributed branched pathways, which we also discussed, bow-tie structure of cell signaling pathways, the Bifan motifs, which is the most common network motif in biological networks. We also discussed that already. Negative feedback loops that are found more so at the membrane in cell signaling networks compared to feedback loops found within the cytosol or later on in lower steps in the network topology. More positive feedback loops than negative feedback loops. And those feedback loops are nested. And now we're going to explain some of those findings in a little bit more detail. So we already discussed the idea of scale-free networks and the interesting observation that real networks are not random Erdos-Renyi networks, but follow this connectivity distribution that fits a power law. And this potentially emerged through the rich get richer evolutionary process or the duplications divergence process or combinations of both of them. And indeed, when you look at various types of biological networks, including yeast to hybrid screen for the yeast proteome, or if you look at the E. coli gene regulatory network, you'll find that most of those networks follow this scale-free distribution, and many other networks in biology, including those molecular networks that we care about in this course, follow that connectivity distribution. Another paper that examined the topology of biological networks identified that there are two types of hubs, party hubs and date hubs. The idea is that highly connected proteins in protein-protein interaction networks can be of either of two types. Those who interact with their partners in one place and one time within the cell, and those are the party hubs, and those other highly connected proteins, but those proteins interact with their partners at different times and at different places in the cell. So those are multifunctional proteins that are reused for various biological functions, and those are the date hubs. Another study that looked at the hub proteins in the protein-protein interactions networks and further analyze properties of those hubs was a study by Mark Gerstein's group from Yale. They identified that there are also two types of hubs, multi-site hubs and single-site hubs. The multi-site hubs, those are also the party hubs, and those proteins are scaffolding proteins that can interact with many proteins at once. The single-site hubs, those are the date hubs, are typically enzymes or other types of proteins that are more promiscuous in their ability to bind to multiple partners at the same binding site. These could be enzymes like kinases and other proteins such as actin that can bind to many pockets of many proteins. What's important about this study is that the structural biology component of protein-protein interactions was also considered in the network analysis. And this is just a review of one of the papers that I mentioned before, and in that paper we analyzed the cell signaling network I'd created from literature. And in that network, if you follow the paths from receptors to effectors, you see that there is also a power law distribution of the number of branches that you can find from various receptors to their effectors. Another property of complex systems that we will find not only in biological systems but also in technological systems is the idea of a bow tie structure. An analysis by Oda and Kitano showed that cell signaling networks are organized in this bow tie structure. So many of the receptors, for example, toll-like receptors, receptor tyrosine kinases, they share the same molecules that transduce the signal from those receptors to their effectors. So although the receptors can be very different at the membrane, there are some adapter proteins and enzymes that are shared. If we talked about G protein coupled receptors, those are the G proteins, G beta gamma, and the various G alpha proteins. If we talk about the toll-like receptors, molecules such as MyD88 could be considered molecules that are shared among those various receptors. And when we talk about tyrosine kinase receptors, molecules such as GREB2 and SOS could be also considered those components that consolidate information and integrate information from various receptors into the same pathways. Once the information is consolidated at those nexuses or those entry points, then the information fans out to affect various effectors, which include transcription factors and other cellular machine components. And this is a review of the network motif paper that was published by Uri alone, and it highlights the Bifan motif that I mentioned before that was probably emerging through duplication divergence, and it's highly enriched in various types of biological networks, including cell signaling networks and gene regulatory networks. So I showed you this network before in another slide. This is the cell signaling networks that we extracted from literature for my thesis. And we found some interesting properties in this network that include enrichment for the Bifan motif, which was highly enriched in this particular network. And to explain why this Bifan motif is so enriched, we looked carefully at what are the types of Bifans. So besides the concept of duplication divergence that generated those Bifans, we looked at the types of nodes, and you can see that the Bifans are most receptors that interact with their substrates, such as the G protein coupled receptors interacting with their G proteins, or other receptors interacting with various adapters. There are Bifans that are created by kinase cascades. There are Bifans that are created by the proteins that regulate the G proteins, such as the GAPs and the GIFs. And also we find a lot of Bifans which are kinases phosphorylating transcription factors, or kinase and phosphatases regulating transcription factors. Another feature that we found in that cell signaling network was that negative feedback loops are more enriched close to the membrane, while positive feedback loops are most enriched once you go more steps into the system. So in order to identify those positive and negative feedback loops, we implemented a network expansion algorithm when we start expanding the network at the components at the ligand receptor interactions. And then as we expand the network in steps from those entry points, we count the number of feedback loops and we look for positive and negative feedback loops. And as you can see at step 4, we found more negative feedback loops than positive feedback loops, and at later steps, or in general in the network, there are more positive feedback loops than negative feedback loops. And this is compared to shuffled networks, where the negative feedback loops and positive feedback loops are relatively even. So this is it for this particular segment. And if we go over those network properties that I just discussed, again, we noticed a power law connectivity distribution in many of the molecular networks. There are party hubs and date hubs, and they match the multi-site and single-site hubs. There is also a power law distribution of branch pathways from receptors to effectors. There is a bow tie structure in the cell signaling network. The Bifan is the most common network motif in cell signaling networks, as well as in other regulatory networks. Negative feedback loops are close to the membrane, and the fact that we have more positive feedback loops and less negative feedback loops, that helps the network to become close to a completely stable system. And this is something we explored in another publication where we talked about monotone systems topology, which I'm not going to go into detail here. So now I would like you to think about complex systems in general, biological and technological, and try to identify this property of bow ties. Can you identify other examples of bow ties in complex systems? And I'm giving you a hint here on this slide, but try to think of examples that are beyond this particular example that I show here.